Hello everyone. So this is a question from RE-NEET 2026. The correct statement about peptides and proteins is okay. So there are a few options given to us. The chapters involved is biomolecules. Okay. So we should know what are peptides and proteins. Okay, important. So proteins are polymers. Okay, in which the monomeric units are the alpha amino acids. Okay, so proteins are polymers in which the monomers are alpha amino acids. Okay, so alpha amino acids can be generally represented as carbon connected to COOH, connected to R, connected to H, which is connected to NH2. Okay, so this COOH is the main functional group, which is the carboxyl group. Okay, and since this carbon is adjacent to the main functional group, this is the alpha carbon. Okay, and this R is the side chain, and this NH2 is the amino group. Okay, and this amino group is on the alpha position, so it is connected to a carbon which is our alpha carbon, so therefore these are alpha amino acids. Okay, cool. So then we have to talk about peptides. So peptides are formed by condensation of amino group of one alpha amino acid with the carboxyl group of another molecule of same or different alpha amino acid with the elimination of a molecule of water. Okay, important. So I will write it down. Peptides formed by condensation of amino group, that is the NH2 group of one alpha amino acid with the carboxyl group, that is the COOH group of another molecule of, can be same or different alpha amino acid with elimination of a molecule of water. Okay, so for example, let's say COOH group of glycine combines with NH2 group of alanine. Okay, so that is with respect to reaction, I can draw glycine as NH2 CH2 C double bond O OH. So this is my glycine. So it is combining with alanine. Alanine is, so I will separate one H from NH2, CH, CH3, and COOH. Okay, so you can see OH and H from here goes out. So we lose a molecule of water and we get our product, which is NH2, CH2, C double bond O, NH, CH, CH3, COOH. Okay, so this is our peptide bond. This COONH is our peptide bond. Okay, so you can see in the definition, so it is peptides are formed by condensation of amino group of one alpha amino acid. So this is the amino group of one alpha amino acid with the carboxyl group of another molecule of the same or different alpha amino acid. So this is the other carboxyl group. So these two guys combine by a loss of molecule of water and they result in the formation of a peptide bond. Okay, so two alpha amino acids combine to give us dipeptide. Two alpha amino acids combine to give dipeptide. For example, this guy. Okay, similarly, three alpha amino acids will combine to give us tripeptide. And if a large number of alpha amino acids are joined together by peptide bonds, they form a polypeptide. Okay, large number of alpha amino acids join to form polypeptide. Okay, and therefore all proteins can be called as polypeptides. Very important. So all proteins are polypeptides. The structures of proteins are discussed at four different levels, that is primary, secondary, tertiary and quaternary structures. Okay, so the first one is the primary structure. So the specific sequence in which the various alpha amino acids present in a protein are linked to one another is called its primary structure. Okay, for example, in hemoglobin, a part of the sequence looks like this. So we have valine connected to histidine, connected to leucine, connected to threonine, connected to proline, connected to glutamic acid, which is connected to one more glutamic acid, connected to lysine, and it goes on. Okay, so this is one particular part of the sequence in hemoglobin. Okay, so this is the primary structure of hemoglobin, which talks about the various alpha amino acids and the sequence in which they are connected to or linked to one another. Okay, and these are the various alpha amino acids. Now coming to the secondary structure, it is the 3D geometric arrangement that the polypeptide chains assume as a result of hydrogen bonding. Okay, so secondary structure is the 3D geometric arrangement of polypeptide chain due to hydrogen bonding. Okay, and the following two different secondary structures are possible. The first one is alpha helix structure. Okay, so intramolecular hydrogen bonds, that is hydrogen bonding in the same molecule, are formed between C double bond O of one alpha amino acid residue and the NH of the alpha amino acid that is situated four residues ahead in the sequence. Okay, important. Intra hydrogen bonds formed between C double bond O of one alpha amino acid residue and NH of amino acid that is situated four residues ahead in the sequence. Okay, to understand this, we will refer to a diagram. So yeah, so you can see here, this is the sequence of alpha amino acids. Okay, so because of hydrogen bonding in the CO of one amino acid, so let's say this is one such sequence of amino acids. Let's say this is one such sequence. So this is the second alpha amino acid sequence. This is the third one. This is the fourth one, and this is the fifth one. Okay, so there is hydrogen bond between CO of one amino acid residue. Let's say this is the first one, and the NH of the amino acid that is situated four residues ahead. So let's say this is the next one, 2, 3, 4, 5. So this is connected to the 1, 2, 3, fourth residue of alpha amino acid in the sequence. Okay, so you can see there is a hydrogen bond between CO and NH. Okay, so because of this hydrogen bonding in the polypeptide chain, the polypeptide chain coil up into a spiral structure. As you can see, they coil up because of the hydrogen bond between this CO and NH, and they coil up into a spiral structure called the right-handed alpha helix structure. So this is the right-handed alpha helix structure. Okay, so be careful that it is the right-handed alpha helix structure. Okay, to understand this, I have one more diagram. It is this one. Okay, so this is the difference between a right-handed helix and a left-handed helix. Okay, so our alpha helix in proteins is right-handed helix. Okay, so in option A they have mentioned that in alpha helix the polypeptide chain is twisted into a left-handed screw, which is incorrect. Okay, the polypeptide chain is twisted into a right-handed helix in the case of proteins. Okay, so therefore option A is incorrect. The second secondary structure that is possible is the beta pleated sheet structure. Okay, so we'll see that using a diagram. Yes, so these are the beta pleated sheets. Okay, so in this the polypeptide chains lie side by side in a zigzag manner. You can see these are the chains, and they lie side by side in a zigzag manner. Okay, and the two such neighboring polypeptide chains, that is these guys, are held together by intramolecular hydrogen bonds. Can you see O and H, there is a hydrogen bond, H and O, there is a hydrogen bond, again O and H, hydrogen bond. So yes, option D, which says that in beta pleated sheets peptide chains are held together by intermolecular hydrogen bonds. Important, intermolecular is between two different molecules. So you can see two such chains are connected by hydrogen bonds. Okay, in the alpha helix structure the hydrogen bonds was in the same molecule, so therefore it is intramolecular hydrogen bonding. In the case of beta pleated sheets, it is with two different chains, so therefore it is intermolecular hydrogen bonds. So yes, option B is correct. Okay, sorry, not option B, option D. Okay, now coming to the third structure of proteins that we discuss about, that is tertiary structure. Okay, so in tertiary structure, it is the overall 3D shape of a single polypeptide chain. Very important. Overall 3D shape of a single polypeptide chain. Okay, that is, it refers to the manner in which the entire protein molecule folds up in the three-dimensional space to produce a compact form. Okay, to understand this, we'll again refer to a picture. Okay, so you can see this is the tertiary structure that I'm talking about. Okay, so this is the overall representation of the single polypeptide chain. Okay, so option B says that tertiary structure of proteins has two or more polypeptide subunits. No, it has only one single polypeptide chain. It tells us how it folds in the three-dimensional space. Okay, so therefore option B is incorrect, because option B says that tertiary structure has two or more polypeptide subunits. Okay, it has only one. Now coming to the fourth kind of structure, that is quaternary structure. Okay, so many proteins, for example myoglobin, exist as a single polypeptide chain. So this guy has a single polypeptide chain. Okay, but there are certain proteins, for example hemoglobin, which exist as assemblies of two or more polypeptide chains. So this guy has two or more polypeptide chains. Okay, so the quaternary structure refers to the spatial arrangement of these polypeptide chains with respect to each other in a protein molecule. So for example, you can say this is the quaternary structure. This is one polypeptide chain and this is the other polypeptide chain. Okay, so quaternary structure tells us about the arrangement of these polypeptide chains in the molecule. So statement C says that only the proteins having a quaternary structure are biologically active is incorrect, because there are proteins like myoglobin and these guys exist as a single polypeptide chain and they don't have a quaternary structure, because to have a quaternary structure you should have more than one polypeptide chain. Okay, so this guy has a single polypeptide chain, does not have a quaternary structure, but it is still biologically active. Okay, so therefore option C is incorrect. Okay, so therefore the correct answer is option D. Okay, in beta pleated sheet structures, peptide chains are held together by intramolecular hydrogen bonds. Okay, thank you.